Good morning and welcome to another great week of instruction. We have completed topic 11, woo, and we are moving on from the bodily systems of animals in general to the body system of the human animal. Let's go ahead and get started. We're going to talk today about human digestion. Okay, so the system. There are two major groups of organs that we are going to use in the human digestive system, and they're going to be defined by how they interact with the food that they're digesting. The alimentary canal is going to be where the food actually physically goes. So it's going to pass directly through these organs, beating a path from the mouth to the anus. Now, if you're having trouble remembering alimentary, let's say that Watson turns to Holmes, Sherlock Holmes, in case you're wondering, and says, "My dear fellow Holmes, I'm finding this case a bit hard to digest. Could you please help me and figure out exactly what it is that I'm missing?" And Holmes turns to him and says, "Oh, of course, my dear Watson. It's alimentary, my dear Watson." Anyway, so and then there's accessory organs, which are not like handbags or shoes, but they so they don't actually transfer food, but they do provide all of the wonderful juices that the food will be digested in, which you guys will be looking into in greater detail later on. Aren't you lucky? All right, so these are the functions of the various different organs in these groups. IB wants you to be able to know their function and how to draw them. That diagram that you're seeing right now is as good a drawing as any. Of course, because the exam is not happening currently, for those of you who are listening during the coronavirus quarantine, you might not have to actually present the skill, but it is required and expected of IB. So I thought I'd let you know. So there are two main methods that we use to digest stuff, mechanical and chemical. And if you're thinking, wow, this actually sounds a lot like when we did DNA extractions, it is a lot like when we did DNA extractions. So the two methods, mechanical and chemical, are going to help break down the food first in a macro scale and then at a micro scale as we get down to the molecular level components of the food. The mechanical, you're going to basically chomp it up in your chompers, and then your stomach's going to turn all over the place, and then it's going to segment up all that food in your small intestine. Chemical is when we're going to have biolin enzymes and all that wonderful acid come in and break it down into smaller pieces. Bile. So we're going to start with chewing. Ha ha! Look at those wonderful photographs. Aren't they disgusting? So believe it or not, there are defined stages of chewing because scientists have nothing better to do. No. So we're going to start with a fancy word for chewing, which means you masticate or grind up the food with your chompas or your teeth. Your tongue pushes that masticated food to the back of your throat as a wet, slimy ball called a bolus. So you're like, "Mwah, mwah, mwah," and a small trap door in the back of your throat that allows air to normally travel to your trachea and lungs closes so that the bolus does not go into your lungs and kill you. This would be called choking. What we can see here on the pictures to the right, this is your, well, I'll start over here with this. This is your epiglottis, which I don't think I actually put here. I think I did, whoops, sorry. The small trapdoor is called the epiglottis, in case you're wondering. And this is the uvula, which is that big droopy thing that hangs down in the back of your throat and that you see in every movie scene with an opera singer. And that stops it from going up your nose, because you don't want your bolus going up your nose, you know? All right, so then after you're done masticating, you churn it into your stomach. So your esophagus squeezes it on down into your stomach. And then as the bolus enters your stomach, all these muscles in your stomach twist and churn, and it breaks down the food even further into a lovely paste. Isn't that a great word for paste? I hope you have wonderful dinner plans. Paste called chyme. And chyme enters the forechamber of the small intestine, so basically the foyer or the opening of the small intestine, where it's ready to be processed. Now, what actually moves it down your throat? Now, believe it or not, it's not gravity, and of course you've experienced this if, like me, you've ever been lazing on the couch and eating food at the same time. Gravity is, I am not pointed vertically down, and so gravity is not pushing the food down my throat. So what's happening is muscular contraction. So longitudinal muscles within your esophageal lining Rhythmically contract to move food in a unidirectional direction. Basically, it squeezes it down your throat like a tube of toothpaste. So if you can imagine holding a tube of toothpaste in your hand and then like, taking your other hand and creating a circle with your finger and thumb and just sort of squeezing that down over the tube of toothpaste to make all the toothpaste move down, that's basically what your longitudinal muscles are doing to move it down your throat and into your lining of your stomach. Okay, so then we have segmentation. So segmentation is going to be a similar muscular contraction, but it's going to happen within your intestines. And segmentation is not unidirectional, it's bidirectional. So we can see here that this time instead of longitudinal, these are circular muscles contracting. And we can see that they're contracting in waves, which peristalsis is sort of like a wave, but we can see it's only one wave and it's moving it just in one direction. But here we have multiple waves, and they're actually splitting up these squares, which are the food, and pushing them in either direction. And basically, what they're doing is they're kind of they're pushing it back and forth. So if you remember when we did the DNA extraction, we put the strawberry in the bag, and then we closed up the bag, and we squished the strawberry all around. That's basically what's happening. We're squishing the food back and forth to make sure that we've gotten all the surface area, we've collected everything that we need before we move it on. Okay, so longitudinal and circular muscles. What did I mean by that? So I just want to clarify that really quick before I move on. When I talk about longitudinal and circular muscles, what I'm talking about is the arrangement of muscles in these tubes, your esophagus and your small intestine. Circular muscles are going to go around the circumference of tubes like that in your body, and longitudinal muscles are going to span their length. Longitudinal muscles, when they contract, are going to make the segment shorter, and when they contract, the circular muscles are going to relax. Now, this is going to seem fairly familiar to you guys if you remember talking about antagonistic muscle pairs before. This antagonistic muscle pair, as the longitudinal muscles, of course, this is in a worm, not in your throat, but we're just going to make this analogy because it works similarly. The longitudinal muscles, as they contract, are going to pull this section shorter, and the circular muscles, as they relax, are going to allow it to open up. So basically, longitudinal contracts, circular relaxes, and we get a larger opening so the food can fit in. After the food is fit in, though, we're then going to go ahead and relax the longitudinal muscles but contract the circular muscles, and that's going to make the tube narrower, and so it's going to squeeze the food along. And so you can imagine how that wave of peristaltic movement is going to travel down the throat and deliver the food to the stomach. In the same way, you can imagine this working in the small intestine. So I just wanted to sort of briefly go back over this. It's kind of cool to see how this actually relates to the stuff we learned about before, because here we have antagonistic muscle pairs. And if you think about it, pushing all the food around inside of the small intestine when we're doing segmentation is basically for the process of reabsorption, which we talked about when we talked about ultrafiltration. So it's pretty cool. Okay, so stomach acid. So stomach acid is an extremely powerful acid. It is released by gastric glands within your stomach. It makes the pH about 2. That is very acidic. Think about the pH scale, it goes from 0 to 14. So 2 is very close to 0, which is the acidic side. So this is going to denature most organic molecules. The lining of the stomach, or the epithelium, is going to contain a mucous membrane that prevents that acid from melting through your stomach. So this is what keeps your stomach intact, even though it's holding this incredibly powerful juice. The pancreas is going to release alkaline bicarbonate ions. Alkaline bicarbonate ions. I can't speak today. You know, you can read, to neutralize the acid. So basically, when all this stuff is finished digesting in the stomach, it's going to want to move on to the small intestine. But you don't just want to take all this acid with this partially digested material and just shove it into the intestine. The intestine doesn't have the protections that the stomach has in its lining. So in order to account for this, your body is going to pump a bunch of alkaline substance or base into that foyer of the inner of the small intestines. It's called the duodenum. It's going to basically increase the pH to around 7 or neutral, so that the fluid that enters into your small intestine will not cause acid or base damage to your intestines. Bile is going to be the other substance we're going to see that's going to be breaking stuff down, and bile is going to be produced by your liver, and so it's going to be related to that limbic system we talked about when we were talking about, back before when we were talking about the immune system, actually. And so we're going to have bile. It's going to be produced by the liver, and it's going to be stored in the gallbladder. Bile is going to contain salts that are going to break down lipids, large lipid droplets, into small lipid droplets, which you can see over there on the picture here. The lipids are being broken down into small droplets. It's called emulsification. And I'm going to increase the total surface area of those droplets so that they can be more easily broken down by enzymes called lipases. So enzymes, we all know, are molecules that help to lower the activation energy of a reaction to occur. They're going to make it more likely that the reaction is going to occur and require less of that ATP. Each enzyme is specific to its substrate, so it's going to bind to either a lipid or a protein or a sugar or something, but it's not going to bind to all of them. And most enzymes that are going to do a lot of the breaking down are going to be secreted by the pancreas. We can see here at the bottom a bunch of examples of enzymes that can be used in this. So we have proteases and endopeptidases, which are going to break down proteins, amylase, which is going to digest a lot of sugars. We can see it's turning starch into maltose there. So starch is a polypeptide. It is a proper polymer carbohydrate, and it's breaking it down to maltose, which is a monomer and is a monosaccharide. Nuclease, which is going to digest in DNA and RNA into their nucleotides, and then lipase, which will digest fats and bile salts, which will help with that, so we can turn these big triglycerides, if you remember those three-tailed beasts of lipid molecules, into monoglycerides. So basically, enzymes are going to be the last step after the macro breakdown has happened. They're going to break these molecules into their micro components. Okay, now we're going to talk about the breakdown of carbs and where this occurs, and the breakdown of all of the macromolecules. So basically, we've covered the organs, we've covered the mechanical and chemical digestion, talking about both the ways that they work, so the teeth, the stomach churning, and the segmentation for mechanical, and the stomach acid, bile, and all that good stuff and enzymes for the chemical. So now we're going to talk about the four different macromolecule groups and where they're broken down. So carbs are going to start being broken down in the mouth. This actually should make a lot of sense because carbohydrates are typically very difficult to break down. Starch is difficult to break down, and cellulose is so difficult to break down that many organisms can't, including us. So starch is going to start to be broken down by amylase in the salivary glands. So while you're chewing, you're going to start releasing that amylase. It's going to break it down in your mouth. The amylase is going to, again, be secreted by the pancreas so it can continue to digest it in the small intestine. So we work on this the whole way through. Disaccharides, which are going to be the smaller little pieces that are going to flake off of the large things like starch and cellulose, are going to be hydrolyzed, so they'll be split apart or split down using water into their monosaccharide components by these enzymes that are immobilized or held steady in the epithelial lining of the small intestine. It's going to be near channel proteins so that the results, those monomers that are produced by that split, can be moved immediately where they need to go. No enzyme can break down cellulose in the human body. It's going to pass through as undigested, which is why people often say to eat your fiber. What you're really saying is to eat a lot of cellulose, because cellulose is going to help to firm up all that material that's going to be exiting you soon, if you get my drift. Okay, proteins are going to be broken down starting in the stomach with the release of proteases. Now, we know that intense acid is going to denature most enzymes. Specialized enzymes that cannot be denatured by very strong acids are going to be released by the pancreas to do this job. The small polypeptide chains that are the results of this initial breaking down in the stomach are going to be further broken down in the small intestine by endopeptidases, which have an optimal pH of 7. So if you remember when we were introducing those bicarbonate ions into the duodenum of the small intestine, we were doing so to make the pH so it would be what we call neutral or normal, and that's where these endopeptidases work. So they work in the duodenum and in the small intestine. They wouldn't work in the stomach because they'd be broken down by the stomach acid and denatured. Lipids and nucleic acids are broken down. So in the small intestine, lipid breakdown begins when bile is released from the gallbladder into the duodenum, where those emulsified fat droplets are broken down by lipase. Nucleic acids are broken down in a similar way by nucleases, which are going to happen also in the small intestine. Every living thing you eat has nucleic acids in it, by the way. So if you're eating something that was alive, nucleic acids are going to be there. And of course, you'll recycle the components to use in your body. So here's a great summary here of the different organs and what is inside of them and what they break down. I think that's pretty cool. And what they produce. So we can see the pancreas is going to be the king of enzymes, liver and gallbladder for bile salts, stomach for proteases and stomach acids, which remember proteases are going to be created by the pancreas but put into the stomach. Esophagus, we have the amylase, and then small intestine, we have our membrane-bound enzymes, which are going to split those sugars down into smaller pieces. Okay, so that's the basic breakdown of digestion. In your follow-ups, you're going to learn a little bit more about it, and you're going to play around with a fun simulation that I hope will help you to sort of get an idea of what each of these pieces do. We'll finish digestion next class with a short little sort of modeling lab, and then we're going to move on to the next step in the human body. And it'll be super fun and also gross because everything to do with the body can be gross. And we're going to make it so because that's fun and it's biology. All right, guys, hope you're staying well and safe. Take care and have the best day ever.